This video begins the second half of Chapter 7, which focuses on synapse physiology. So we'll begin with discussing the neurotransmitter acetylcholine and how it can activate its receptors. So neurotransmitters can activate receptors to open ion channels in either one of two ways. The first way is the direct way. This is where the ligand, being the neurotransmitter, can directly bind to a gated ion channel. The second mechanism is indirect, and this involves the a transmembrane protein being a receptor for the neurotransmitter, and then the activation of a G protein or cyclic AMP activated channels. That would be a second channel that would be activated indirectly by the neurotransmitter binding the receptor. So there'd be a minimum of two proteins involved, one to be the receptor for the neurotransmitter and a second one to be the ion channel. So for acetylcholine, we have two of these type of receptors in the body. We have nicotinic acetylcholine receptors. These are the direct type. These are the ion channel linked receptor. These can be stimulated by nicotine. This is where the name came from. That is the agonist, in addition to acetylcholine. And these are found in skeletal muscles and other parts of the autonomic nervous system, which is the one our body automatically does without consciously thinking about it, and is also found in parts of the central nervous system. The indirect acetylcholine receptors are called muscarinic, and these are G protein linked receptors. So they got the name from the chemical muscarine that can act as an agonist to stimulate this pathway in addition to acetylcholine. And this is found in the central nervous system, as well as smooth and cardiac muscles, and also glands that are regulated by the autonomic nervous system. Again, that's the kind that's happening automatically without requiring us to think about that. So an agonist is a drug that can stimulate a receptor. And I already mentioned that nicotine can be stimulating the nicotinic acetylcholine receptors, and muscarine can be activating the muscarinic acetylcholine receptors. An antagonist, in contrast, is a drug that can inhibit a receptor. So a known inhibitor of the muscarinic receptors is atropine, and a known inhibitor of the nicotinic receptors is curare. So I'd like to take a close look at the nicotinic acetylcholine receptors. So again, this is a direct ion. So here's a receptor in the membrane. This is the open receptor. This is a closed receptor. So the receptor is closed until the acetylcholine neurotransmitter can bind it, and we need two molecules of acetylcholine to bind the two binding sites in the channel. So the channel is closed until the neurotransmitter binds to it. Okay, after the two neurotransmitters bind to it, the channel will open, permitting the diffusion of specific ions. In this case, this nicotinic acetylcholine receptor allows the passage of two specific ions. One is sodium, and the second is potassium. So when it opens, sodium will rush into the cell following its concentration gradient, and then potassium will go out of the cell following its concentration gradient. The effect of sodium on the membrane potential is greater than the effect of potassium, and therefore there's going to be a depolarization event in the cell in this area right under the nicotinic acetylcholine receptor. So you need to imagine that we have tons of these receptors in the membranes of our nerve cells, and depending on how many neurotransmitters there are, and that will determine how strong the signal will be for depolarizing the membrane potential. And we'll determine will it be strong enough to trigger an action potential. So these excitatory postsynaptic potentials, as they're called, can basically depolarize the area. Let's say this is happening in the dendrites or cell body. So here we see, here's one excitatory postsynaptic potential happening. There's another one, right? And this neither of these have made it to the threshold. But this third one, with more neurotransmitter, has enough depolarization to trigger an action potential because it hits the threshold of -50. Then action potential could be fired, and this would have to happen, again, in the axonal hillock, and then the action potential will be transmitted down the axon. So I just want to take a minute to compare and contrast excitatory postsynaptic potentials and action potentials. So this is really important. So if we look at the stimulus for opening the ionic gates, in an action potential, it is depolarization. In the excitatory postsynaptic potential, it would be the excitatory neurotransmitter, for example, acetylcholine. What is the initial effect of the stimulus? Well, for an action potential, the first thing we know that happens is when the depolarization is hitting the threshold, the sodium channel is open. What will happen with the EPSP is that a common channel for both sodium and potassium will open. What about repolarization? Well, we know in an action potential, repolarization will happen from the opening of the potassium gates, but then in the EPSP, it's just a loss of the positive charges with time and distance. So these EPSPs don't travel that far unless there's, well, they don't travel that far, but you have to have a lot of them in order for the depolarization to travel to the axonal hillock. Then it would be traveling far, but you really need a significant amount of depolarization. But with a little depolarization, it won't be hitting the axonal hillock. And this would typically happen right in a dendrite or even the cell body of a cell. What is the conduction distance? Well, in an action potential, it can go it's regenerated over the length of the axon. In the EPSP, it's typically just 1 to 2 millimeters. It's a very localized potential, and you need this to be occurring in so many places and so strongly to eventually get it to be hitting the axonal hillock. Positive feedback between depolarization and opening of the sodium channels. Yes, that happens in the action potential, but it does not happen. There is no positive feedback between depolarization and opening of the sodium gates with the EPSP. What's the maximum depolarization? Well, in action potential, it can go up to plus 40 millivolts. In an EPSP, it will be close to 0. Summation? No, there's no summation in action potential. It's an all or none event. However, in the EPSPs, summation does play a role. It's adding up all these little EPSPs together that can produce this graded depolarization. For a refractory period, there is one for the action potential, and there is no refractory period for the EPSP. And effective drugs, well, acetylcholine effects are inhibited by the tetrodotoxin, but not by the curar. And then, in contrast, the EPSPs, the acetylcholine effects are inhibited by the curar, but not by the tetrodotoxin. And we'll talk more about those in a little bit. So the second kind of acetylcholine receptors are called the muscarinic, and these are indirect. In this case, we only have one acetylcholine molecule that binds onto the receptor for the muscarinic acetylcholine receptor. When it binds to the receptor, what will happen is there will be an activation of the G protein. So just to remind you that the alpha subunit is typically holding on to a GDP. It'll attach to G. It'll then have a GTP attached instead of a GDP, and when the alpha subunit is binding a GTP, it separates from the beta and gamma subunit. Those subunits will separate. In this case, the beta and gamma subunits of the G proteins will travel in the membrane to the ion channel. And in this case, the ion channel for the muscarinic receptor is a potassium channel. And in this case, the potassium is going to move out. This is going to cause a hyperpolarization or a more negative resting membrane or a more negative membrane potential. So the resting membrane potential is negative 70. It'll be more negative, and that means it's less likely that this nerve will be firing an action potential. We call this an inhibitory postsynaptic potential. And where is this important in the body? So in the heart, the potassium channels are opened, creating these IPSPs through the hyperpolarization. And that is because acetylcholine, what it does in the heart is it opens the potassium channels. I need to disclose now a complication in our human body, and is that sometimes acetylcholine muscarinic receptors in some parts of the body can actually close the potassium channels. And that would be an excitatory postsynaptic potential. So where does that happen? So that happens in smooth muscles of the stomach. Then there, the potassium channels are usually opened, but acetylcholine binding to the muscarinic acetylcholine receptor will actually lead to the closing of the channels via the G protein mechanism, and this will lead to the EPSPs and basically lead to the depolarization and contraction of these muscles. So what happens after acetylcholine is released into the synaptic cleft, right? So we know that there's going to be signaling of either in this case, it's the muscarinic acetylcholine receptor, because we see only one acetylcholine in the postsynaptic cell. In the presynaptic cell, we see it has secreted acetylcholine. And I want to remind you that's going to happen from the vesicle containing acetylcholine, fusing with the plasma membrane, and then leaving via exocytosis. So you can imagine once an acetylcholine binds to a receptor, triggers the opening of the ion channel, that then is going to detach from the receptor, but then maybe it can bind it again, right? So the body has a mechanism to prevent overstimulation by neurotransmitters. And in the case of acetylcholine, the postsynaptic cell has an enzyme in the membrane called acetylcholinesterase. Acetylcholinesterase has the ability to hydrolyze acetylcholine into acetate and choline. When acetate and choline are separate, they no longer can interact with the receptor and trigger the ion channel opening. And then after acetylcholinesterase in the synapse has basically breaks acetylcholine into acetate and choline, then the acetate and choline can be taken up again by the presynaptic axon. And then it can be recycled to be reused again. But with this, acetylcholinesterase really plays a key role in preventing overstimulation of the postsynaptic cell.